Okay, so it's 2 o'clock, so I'm gonna start recording, and I can start with introductions, and hopefully more people will log on, but we have 13 on so far. Welcome to the first IWiMS Clinical Trials / Research Group Conference, and we're hoping to have people from different areas. So we have definitely Australia is represented, and the US. I see some from Canada. And so hopefully more places will be represented. So today in this conference, we have 3 great talks. So we'll start with Dr. Ellen Mowry from Johns Hopkins, then Dr. Riley Bove from UCSF, and then Dr. Walt from Monash University in Australia. The conference will be recorded. You can also, if you want to ask a question, you can ask a question, or you can also send a message through the chat box, and then I'll monitor that. And the talks will be about 15 minutes with time for questions afterwards at the end of each talk. All right, so we can get started. So I will stop sharing, and Dr. Mowry can start her talk. So the talk is Investigator-initiated Clinical Trials in MS, the good, the bad, and the ugly.
Excellent. Thank you so much, Kristen. And I thought I would just kick us off with a talk that hopefully goes through trials more broadly, and for maybe more junior people, we could essentially review some of the pros and cons of doing these talks and doing these trials and whatnot. And I'll tell you a little bit about some of the results of some of our pilot trials. So these are my disclosures. So I thought I would just talk a little bit about how I got interested in clinical trials, because I feel that Emmanuel's laughing, that these
I recognize the
don't necessarily require that you start off as a trialist, or I don't know, maybe that at least when I was before meeting Emmanuel, was always of the impression that you had to fit in a certain niche in your career and you had to be known as the X person. And I feel like personally that diversifying one's experience is optimal. It's A, more fun, and B, kind of allows more flexibility in one's career to maintain academic success. And we'll talk about that a little bit further. But anyways, I kind of got interested in investigator-initiated trials when I was working with Emmanuel as a fellow. I was doing a lot of clinical research, and of course Emmanuel is well known for not only doing great trials, but also for having really excellent cohorts. And I started getting interested in modifiable prognostic factors for MS. And we did an initial study of vitamin D levels in children with MS, showing that they were inversely associated with relapse risk. And so I applied for a Career Development Award to do a similar follow-on study in adults, looking at the impact of vitamin D levels and their association and observational cohort at UCSF with MRI outcomes, essentially both inflammatory and neurodegenerative outcomes. But in a classic K award, you also kind of have a little pilot trial as your aim 3 if you're doing a clinical research. And it seemed natural to do a little pilot trial looking at vitamin D response to supplementation in people with and without MS, because clinically we were noticing that a lot of people with MS, when we were giving them vitamin D, weren't really bumping their levels as we would expect. And we were wondering if this was part of the disease, essentially. And so that K was successful. And then during that fellowship, I was doing a bunch of coursework in clinical research, and one was on clinical trials, and we had to write a clinical trial protocol as part of the course. And I realized we should probably actually do a clinical trial. And so I just sort of submitted it, and the funding was successful. And around that time, when it was about to start, I moved back to the East Coast. As Emmanuel and I say, we had a divorce, a very sad divorce. But since then, I've done a lot of investigator-initiated trials, both little pilot studies as well as pretty large multi-site trials. And so I feel like I've learned a lot, and each trial I design, I learned something from, both something great and positive as well as what not to do the next time. So I thought I'd walk those of you who might be interested in doing trials through just some basic overviews, and others may have recommendations as well with respect to how to think about designing your own clinical trial in MS. So I have learned throughout my time in doing trials that there are some really basic tenets that are pretty critical to the success of your trial. And some of them seem really obvious, but I think are worth thinking about. So first is choose eligibility criteria wisely. My first trial, that vitamin D pilot that I did, had about 20 criteria for entry that were really stringent. And it made it really difficult to find patients as well as controls in the case of that pilot trial for the study. Now, it kind of needed it for the type of hypothesis we had. We were looking for different responses to vitamin D supplementation. So we really want to be careful to make sure there was no impact of differential, skin pigmentation or body mass indices or things that might otherwise be responsible for any differences that we found. But the downside was that it was really hard. I had to comb through a lot of clinical charts, and at the beginning anyways, these were all paper charts. So it took a lot of time to find participants for the trial. And then, when you're thinking about the trial population you're studying too, of course, I think one of the things that we know about a lot of our large pharmaceutical trials in MS is that the populations that are included in those studies are not necessarily representative of the larger MS populations. Many of the clinical trials have pretty stringent criteria for relapse or MRI activity or degree of progression just to get into the study. And the patients tend to be a lot more healthy than the average population. So that means not only if you make your own trials, the criteria that stringent, will be harder to find people in the real world to participate, but also the results of your study may not apply to a large number of the patients. So you got to really think about whether you want a very stringent trial or whether you want something that's a little bit more generalizable to the population. Another example in the US, for people in the US anyways, is that, less than 1 or 2% of pharma trials have included Black or African American individuals in the trials. Yet a huge chunk of our patient population is not Caucasian, and most of the people, at least in our centers and many others, are Black or African American. So it's something that we really want to, think about carefully, and you may even want to enrich your trials for those individuals. In the US, one thing I learned early, which is a bonus and a curse, is that standard of care activities can be billed to insurance. This is awesome in many ways. So MRI scans are usually hugely impactful for an MS clinical trial in terms of budget, and sometimes can make your trial not feasible. On the flip side, if you're billing MRIs as standard of care, i.e. the ones you're going to get anyways, it means that you have to be willing to do the MRIs at the times you would normally be monitoring people anyways. And so that may not line up with your study endpoints. And then it can be a little bit challenging to get the MRIs standardized fully, because people in the US have differential insurances that may or may not cover MRIs being done at a single center. And it's not really allowed by newer Medicare rules to bill most of those MRIs to insurance, but cover it through your research budget for a few people. So that can be a little bit tricky. So it sort of depends on what you're doing. For example, our large PCORI trial uses standard of care MRIs because there's no way for a 900-person trial we would be able to afford the MRIs that are necessary for a 4.5-year study. However, we made it a tertiary endpoint, recognizing that the data may be noisier because they're not all getting done on a research MRI scan, or people may change insurances and not be able to have the MRIs done on the same scanner. We do standardize the MRI images, however, which is doable, and most radiology centers seem pretty happy to do so. Setting an appropriate balance between collecting enough data and making the visits too onerous for participants can cause challenges with respect to follow-up. And I tend to now make visit windows for the study pretty broad, so we don't have to report a bunch of protocol violations to the institutional review board every year. Kind of think about if you have somebody who tends to cancel or switch their appointments, we always try to book them at the beginning of the window, so that if they're going to flake and have to reschedule, we can accommodate them within the window. And then At the same time, you don't want to not build in some enriching assessments. Maybe you have some other hypotheses that you could test observationally in the study, and you want to kind of use the gathering of the data to your full advantage. So if you're interested in diet and you're doing a study of some other intervention, you may still want to build in some observational data collection of diet so that you can, gather preliminary data for your next trial or next study. I also am really careful telling the IRB what I want to collect in terms of adverse events. For example, almost always now, I try not to say that we're going to collect any adverse events that are known and reported associated with the platform disease-modifying therapy that the person's on, because I don't want to report on those, and it's ridiculous. I never call MS relapse or hospitalization for MS relapse an adverse events. First of all, it's usually an outcome, so it shouldn't be an outcome and an AE. And secondly, I expect that some people are going to have MS relapses and hospitalizations. So, the more you do upfront to make your protocol work for you, the less painful work you're going to have to do along the way. Having a great study team is really crucial. A study manager for me has been life-saving. Ideally, a statistician who can help you. You may need an investigational pharmacy and a coordinator. And so when you're writing your trials and submitting grants, you are going to need to make sure that you budget accordingly and with enough effort. And then prior to doing your study, and maybe even around the time you're writing the grant, you're going to have to think about whether you need to approach the regulatory agencies in your country for an investigational new drug or device application. If you're doing multi-center trials, subcontracting always takes longer than you think. We've started using a central IRB, which has pros and cons. It means all of the documents are sort of within our control, but it also adds a lot of extra work for our central study team, and it's pretty expensive. For multi-center trials, I would say no one's motivated to enroll as you are. And so you have to add a lot more sites than you think are necessary. And then I also say, remember this, if you're the site PI for other people's trials, try to be a good citizen as well. And then in terms of budgeting for other sites, I always think it's appropriate to provide some startup funding, but then make your other payments contingent on enrollment so that you're not spending out your money all at one site that never ends up enrolling, and then out of money to provide to the sites that do enroll. During the trial, I found that I have to be really heavily engaged in order to make the trial succeed and to understand where the pinch points are for other investigators, and to make any changes needed in order to accommodate those. It's important to keep up your clinicaltrials.gov or other equivalent regulatory reporting. Important to kind of hold regular meetings, both with your centralized study team, and sometimes, if you're doing multi-center trials, we've found it really successful to have monthly webinars for that as well. You may want to have some plans to approach sites one-on-one in multi-center trials through various mechanisms to encourage enrollment. We really prefer to monitor data in as close to real time as possible. We found that it's not super awesome when the study coordinator has left 2 years ago and you're trying to monitor handwriting or data entry error. And I personally like, when possible, to have some social events for trial members at ECTRIMS or other meetings. What about the impact of clinical trials on your success in academia? These may apply only to the US, but where we're sort of forced to get a lot of our own funding to cover our salary. But I'm assuming some of these thoughts also apply in other places. So I think the pros of doing trials are, it's a great mechanism if you don't have cohorts that are established at your university to get some projects underway. It takes so long to build a cohort and to start to publish off of it that I found when I moved to Hopkins, that it was really great to have the trial that I had funded at that time to fall back on. I like that the data collection is a little bit more rigorous than a traditional cohort in some ways. And I think it's fun that the trials have some direct impacts on clinical care. Cons, of course, are that it's expensive and very labor intensive. It does take a while to obtain your results, and there are some limitations with the number of questions you can address in any one given trial. So everyone says, well, why do you keep doing this? And I always say it's like a moth to the flame. So here's the results of the first trial that we had. This very tiny little pilot trial, but we had this hypothesis that vitamin D response to supplementation was more limited among people with MS compared to healthy individuals. And at least in this trial, which enrolled white women with MS who had restricted body mass indices, and we enrolled them only in the winter to try to remove any effects of seasonal behavior differences, that indeed these women actually did have a less robust response to vitamin D. And we, it's exciting to see our hypothesis played out. And then our vitamin D trial, which is my very first learning trial for a multi-center study, took forever to enroll because of some of the decisions I made early on, but we're waiting for last patient out. So we're really excited to have those results. I also love trials because you can turn epi results into an answer that's clinically meaningful. So we know, of course, obesity increasingly recognized as a risk factor for MS, less clear if obesity is prognostic. And so despite that, we know that obesity-related comorbidities are associated with worse prognosis. And so we got really interested in this and decided based on those data, as well as some EAE data suggesting that calorie restriction and fasting may lead to less severe course of EAE, that we would try to do a calorie restriction pilot. And so we designed ATTACK-MS, which was essentially looking at sort of intermittent calorie restriction, so eating normal amount of calories 5 days a week and 2 days of very low intake. Continuous calorie restriction, which was the same percentage of calories removed but spread out over all 7 days per week, or control. And we sent food to people using a standardized diet for 8 weeks from the USDA. And we collected a bunch of clinical and blood outcomes. And so you can see that we enrolled 36 people. We had pretty successful completion of the 8-week protocol. And the primary outcome was looking at metabolism, actually. And I won't go into great details, but we saw really interesting changes in metabolites associated with functions that you would expect to think are meaningful, either for immune system functioning or for neuronal health. And moreover, we saw that the weight loss groups, the calorie restriction groups, had meaningful improvements in depression. So to me, this is a really great talking point for patients. I say, like, I don't know what's happening with your specific MS outcomes, but I can tell you that if we can get some weight down, you will probably feel better. And it may have some interesting metabolic effects. I also think it's exciting that our trials could impact day-to-day care. And also the patients that I see tend to continue to inspire me about like what matters most to the patients. And so I'm always coming up with new ideas when I'm seeing patients. And the trials themselves give me new ideas about what might be the next appropriate study. So for example, that calorie restriction trial that I showed you, after that 8-week period, the participants were supposed to follow a calorie restriction diet on their own without getting food shipped, and they sort of tanked. So it inspired 2 new clinical trials to try to address why are patients unable to adhere to behavior change. The questions that you're answering or addressing are kind of using the best kind of design to remove confounding and bias. And I hope that every trial you design kind of is better than the one before. And you get to work with a team, and it's fun and dynamic, and you feel like you're in a big community. So I think I'll end there. Happy to take questions. These are some of the many people at Hopkins, throughout the rest of the world who helped, and I'm grateful. Thanks.
Thank you very much for that talk. And I think that was a good introduction to our clinical trials group, pros and cons, and hopefully motivating some people to start their own trials. So anyone have questions for Dr. Mowry, comments from your own experience? And if you do have a question or comment, it would be good if you can just introduce yourself and where you're from.
So, Ellen, great talk. I have a question for you in terms of funding, do you want to talk to the group about, places where you look for funding when you're trying to design a clinical trial, knowing that traditionally women have not been well represented in the pharma-sponsored trials, and that's something IWiMS is actually trying to address, we're going to work directly with companies about that.
Yeah, I mean, I found funding to be, painful as always, but there are always more opportunities than I realized. So I think the National MS Society has been a good funder of some of the research. It can be challenging. It depends a little bit on the trial you're doing, because obviously they're not the NIH. They don't have the enormous budget that the NIH or other institutions may. And so trials cost a lot of money. I probably should have put that as a con. I think I did, actually. And so usually the NMSS, for most studies, they kind of cap their direct costs somewhere around $200K per year. And unless there's some really strong impetus to fund something outside of that range. So that really does change kind of what's available. So you could either try to see if you can create a study within, reasonable sample size and cost low enough to meet that, or you may have to look outside. I think in the US, the National Institutes of Health, the Neurology Institute, NINDS, actually it's pretty hard to get funding for clinical trials. They have an exploratory clinical trials kind of pathway, but you have to kind of meet certain criteria, essentially a phase 2 trial that will generate go, no-go decisions about doing a phase 3. And then there's NeuroNext, which also has very specific criteria. I have found, though, that there are some other institutes that are very interested in clinical trials that can span across different disciplines. For example, the National Institute of Nursing Research actually has quite a few clinical trial mechanisms that could apply broadly. And so that's useful. We have funding from PCORI, the Patient-Centered Outcomes Research. The Department of Defense often has funding opportunities. And lastly, pharma has a lot of good mechanisms as well. Investigator-initiated trials. I have one pharma-sponsored trial. And that actually, I think when you pitch something to pharma, it's helpful to think about, you don't want to, sacrifice any of your own integrity in doing the study, but it's always helpful to think about, well, what could be in it for them, right? You have to point out where the hole is that you're trying to fill that actually matters to the pharmaceutical industry, and pitch it like that. And actually I found them to be pretty responsive to that sort of pitch, and thus very interested in funding, I guess, not only trials, but even observational pilots or other things like that, which might give you either the trial you want or the pilot data you need to apply for a larger pot. I don't know what others do, but that's sort of my strategy.
Great, thank you very much. All right, so I think we should move on to the next talk. So this is Dr. Bove, so if you can share your screen. So Dr. Bove is at the University of California, San Francisco, and she'll be presenting on her pilot trial of hormone therapy for women with MS.
Hi, everybody. Okay, so this is sort of work and analyses. We just completed this trial, and sort of the analyses are ongoing. So this is just a very, very preliminary, fresh look at the data. So welcome any and all input here from people who have a lot more experience in the trial arena than myself. These are my disclosures. And this was a pilot trial that I started right at the beginning of my sort of junior faculty period, right when I transitioned to UCSF from Boston. And so just to give a little bit of background of why I was interested in looking at this topic. So when I was just starting my fellowship, very, very little was known about the menopausal transition in women with MS, and it's probably because around the time of the menopausal transition, MS starts to progress. So various groups have shown that sort of age 45 or 50 really seems to be the time where there's significant progression. And so in the pre-DMT era, by that point, women were quite impaired. And so the question of menopause really wasn't asked. So we started to look at it a little bit, and we looked at the age of natural menopause in 2 separate cohorts, and we found that it was right in line with the median age at natural menopause in women living in Western populations, so 51.5, which is just quite typical. And this was the reason we even asked this question is because some women with neurological conditions like epilepsy actually have an earlier age at natural menopause, and whether that's simply a drug effect or whether it's because of the effect of seizures, on hormonal regulation, it's uncertain. But we wanted to at least ask the question. In terms of what happens with the menopausal transition after menopause, again, in 2 separate populations, one looking at a patient-reported disability scale, and in the other population looking at physician, reported EDSS, recorded EDSS over time, we found that at the time of the final menstrual period, you see that the EDSS slope does significantly accelerate. And this is sort of independent of chronological age. Several other groups have looked specifically at menopause before and after, and both relapse rate and disability progression reporting in one group. Can you guys see the full screen here? Okay, in one group reporting that the relapse rate after menopause did decrease, but EDSS did not really change. Another group actually did report that also a decrease in relapse rate after the menopausal transition, but also an acceleration in EDSS, as we had found. In addition to disability progression, at around the time of menopause seems to be when a lot of patients will tell me sort of just, in visits that they feel like things are falling apart or they can't keep it all together. They're really sort of experiencing sort of a jumble of symptoms and really need help kind of teasing apart the effects of just normal aging, of multiple sclerosis, of life changes like changes in career or dissatisfaction with their marriage or empty nest or things like that, and then menopause itself and the effect of menopause on MS. Sort of one classical symptom that I hear is that the hot flashes, so the vasomotor symptoms, really seem to kind of trigger. I don't know that it's a UTAS phenomenon or whether it's sort of a separate physiological phenomenon, but when women are having hot flashes, it really does seem to trigger kind of a worsening in their MS symptoms. In addition to that, changes in mood, cognition, sexual function, etc., are really sort of strong overlap symptoms. What do we know about hormonal therapy in MS? We really know very, very little. So most contemporary cohorts have very low rates of use. So women sort of followed after the Women's Health Initiative really have very low rates of use, less than 20% in cohorts that we've looked at. And so it's very hard to sort of get good, even observational data. In addition, there's probably a large bias in the historical cohorts in the sense that women who were doing better were more likely to get age-appropriate care that included hormonal therapy at that time. And we all know that there's a big mismatch between observational and interventional studies in general, and specifically with relation to hormonal therapy in the general population. So observational studies have suggested that hormonal therapies have a good effect on cognition, and interventional studies really haven't shown that to the same degree. So we did look at the Nurses' Health Study when I was still a fellow. We looked at sort of, again, this cohort of women who went through menopause at a time when women were actually being advised to start hormonal therapy. And we found 95 women with MS, and we looked at their first postmenopausal time point. And we found that the women who were on hormonal therapy reported significantly better physical function, so PF10 scores, than women who had never been on hormonal therapy. The catch was that when we went back and looked even at their premenopausal time point, the women who went on to receive hormonal therapy at menopause were already doing better. The numbers are very, very low here, but it does suggest again that it's the women who are doing better who are put on hormone therapy, and not that hormone therapy necessarily helped their physical functioning. So the aims in the pilot trial, knowing that basically we had almost a complete lack of data with respect to hormonal therapy in menopausal women with MS was to see whether a type of hormonal therapy was well tolerated, improved vasomotor symptoms, and also MS-related symptoms and quality of life. And so just simply, we compared hormonal therapy to placebo, looking at tolerability, menopausal symptoms, and MS symptoms. So the methods, it was a single center, randomized, controlled, double-blind, phase 2 clinical trial. It was 8 weeks of treatment with a tissue selective estrogen complex versus placebo. I'll talk about that in a second, but I don't know if someone had a question. Okay. And the participants' major inclusion criteria were MS by immunomacdonald criteria, age 48 to 55, amenorrhea at least 6 months, and hot flashes that were bothersome. And the major exclusion criteria were relapses or steroid treatment in the prior month, contraindications to MRI, estrogen therapy, angioedema, or current hormonal therapy. Just to talk a little bit about the tissue selective estrogen complex, what it is, it's an estrogen plus a SERM. And the rationale is that you want to give an estrogen, but you also want to protect the uterus, stabilize proliferation. When you give estrogen plus progesterone together, you can increase the risk of certain cancers. And therefore, the SERM, which is selective, which selectively antagonizes, this specific one antagonizes the effects of estrogen on breast and uterine tissue, you can actually give estrogen to women who have a uterus without having to use progesterone. And so, again, the SERM is BCEA or bezadoxifene. And it was approved by the FDA in 2013. So this is the sort of schedule of study activities. And basically after a screen, there was a visit 1, and then patients went home and took either the drug or placebo, randomized 1-to-1, and then returned 8 weeks later. And just 12 of the patients also underwent an MRI. The decision to undergo an MRI was really patient-driven, and so we ended up not having balanced groups. So 4 women randomized to placebo and 8 randomized to drug underwent an MRI. And one of the sort of things I wanted to bring up for the conversation, for the discussion part, was that because we knew we had just an absolutely low sample size, we were absolutely unpowered to look at anything statistically significant. And we knew that because just the planned number was 24. It's very, very low, but also because there's a huge placebo effect in hormonal therapy trials. And so even more than other types of trials, you need a very, very large sample size in order to counteract the effect of the placebo effect. And so we worked with our statistician at the Brigham to come up with what he called the play the winner approach. And we basically had 80% power to observe a greater mean change in the treatment arm than in the control arm. And that was all it was. And so basically we just wanted to tabulate, did we get more points for the drug or for the placebo? Just to show you the table 1 at baseline, 12 patients were randomized to placebo and 12 to treatment, and they were matched on everything in terms of age, disease duration, EDSS, a range of measures, and there were no significant differences in the DMTs. So, in terms, actually, no, that's right. In terms of the results, so here's sort of the general categories. So tolerability, menopausal symptoms, and MS-related function, quality of life. In terms of study completion, so one patient in the treatment arm took her study drug and then just never came back for visit 2. She lived in Hawaii, actually, and just didn't fly back. In the placebo arm, one patient discontinued because she felt that there was no effect on her vasomotor symptoms, and those were so bothersome and severe that she wanted to go on sort of regular hormonal therapy. And one patient unfortunately died from unrelated causes. It was undisclosed substance abuse, and died of polysubstance abuse. In terms of missed doses, fewer treatment patients missed any doses. There are fewer GAD-enhancing lesions at week 8 in the treatment arm. There's better treatment satisfaction in a global score. At the end of the study, the women in the treatment arm had fewer hot flashes. They had better sleep quality. And then also in terms of looking at changes in MS-related outcomes in the treatment arm, women had more decline in EDSS. The only difference where the placebo arm did better was on quality of life physical change. Otherwise, all the other ones, time 25-foot walk, MSNQ, SDMT, Z-score change, etc., the treatment arm did better. So I just wanted to, first of all, thank all the coordinators, the study statistician, and the referring clinicians, and then and funding. So this was funded through a National MS Society pilot trial. And then wanted to go back to the discussion and sort of, talking about a few things. One is the obvious limitations of very small sample size. This is really just a pilot just to make sure that we weren't seeing any dramatic effects, for example, on inflammatory lesions in this age group, because so little is known in general in this age group. And then, to sort of elicit any thoughts that you all may have about the sort of interpretation of results, and so specifically the statistical play the winner approach. That was a pre-specified approach that we actually wrote in our grant way back when we formulated the trial, but it's not sort of familiar necessarily to many people. And so would love your perspective on that. And then sort of on next steps, do we think a bigger trial is even necessary here, even feasible?
Thank you very much for going over that trial. Does anyone have comments about these topics? I think they're very helpful to discuss.
I have a question. I'm Riley Anneke Vanderveld here from Melbourne in Australia. Did you struggle to, I find that whenever I even mention hormone replacement therapy, there's such a stigma around it, and it caused cancer and all these things. Mm-hmm. That I would have imagined that recruiting for a trial like this, even with a different SERM molecule, would have been quite tricky. And I wonder if that's also going to inform your decision to do a larger trial.
Mm-hmm. I think you're absolutely right, Anneke. And when it was I remember Joanne Manson, who sort of wrote up the initial Women's Health Initiative, sort of negative trial results. She came to speak when I was in medical school, and I just remember that just like put the kibosh on hormonal therapy for a long time. Subsequently, of course, re-evaluating the Women's Health Initiative results, they've really found that actually it's really when you gave the hormonal therapy to the much older age group where you saw the really negative effects with respect to brain health specifically. And so I think there's a general trend in reevaluating the literature to not be as apprehensive about using hormonal therapies if there are clear symptoms, and if you're clearly targeting something like vasomotor symptoms, and if you're clearly giving the lowest dose you can for the shortest time period to tackle the symptoms. So I think there's, and it was interesting when I moved from Boston to UCSF, both groups have been very heavily involved in hormonal therapy trials, and the UCSF group was much more apprehensive in general. And I think people are coming around again to the idea that it may be useful and helpful, not as dangerous as sort of, we thought 5 or 10 years ago. But that did cause some difficulty with recruitment because some clinicians were uncomfortable. And so, we'd always say, I'd like to talk to your primary care doctor or your OBGYN and your MS doctor and make sure they're on board with you enrolling. And then we had to deal with questions at that point. Yeah.
Did you have any specific exclusions around, excluding women with, say, hypertension or, family histories of breast cancer? Or did you mandate any sort of screening for these things before people entered into the trial?
Yeah, so because most of the women were entering after the age of 50, so many had had a mammogram, a negative mammogram, which sort of we wanted to get the records and just sort of make sure. So that was one point. And then any personal history of breast cancer, we absolutely excluded. The interesting thing is actually the SERM is being tested separately in trials to prevent breast cancer and to treat breast cancer, sort of like tamoxifen. So, you know, our breast cancer experts here at UCSF are actually really positive about the drug. So any personal history, yes, and then familial history was on a case-by-case basis.
Riley, I have a question or comment. Hi, great job. And I'm really impressed with how much you got done on a pilot trial budget. It's like not a huge budget for those of you who don't huge budget trials. So it's really impressive. I mean, having done a few pilots myself, I think, you know, when we power studies, we're always making some sort of assumptions about effect sizes that are literally just assumptions. So of course, reviewers are looking for some magic number, but in reality, they're just assumptions. And you could argue that, you know, the thing you're really interested in is, does this affect progression? You didn't see any danger signal. Mm-hmm. Your assumptions about the rate of progression in this 8 weeks, you know, you're not going to see any difference. Mm-hmm. Maybe one thing you could say is it wasn't really intolerable for people. You didn't see any major danger signals, you know, in terms of ability to take the medication for the most part. And, you know, maybe it would be worth doing with really careful, or thinking about with very careful, you know, stopping rules to do a larger trial, right? Mm-hmm. Progressive MS really stinks, right?
That really stinks. I will say the SERM specifically, actually, and I think you know this, Ellen, but we just published basic preclinical data showing that the SERM actually works amazingly to promote myelin repair.
Yeah.
Through non-estrogenic mechanisms, which
right.
was the thing. I was convinced, you know, that this SERM is going to be great, and if you've read the data on estrogens, you know, they do a wonder for myelin repair. So we tested it, and it did great. And then I was sort of ready, you know, okay, we proved our point, and so, you know, the myelin expert, Jonah Chan, said, well, you have to prove that it's through the ER receptors. So it took us forever to breed the ER double knockouts, because obviously without any estrogen receptors, it's very hard to breed. But we got the ER double knockouts, and then we gave them the SERM, and it worked just as well.
Cool.
That it's totally estrogen independent effects.
Yeah.
The fact that it also works on estrogen. So we have patient number 6 enrolled today in the myelin repair trial. So that's really exciting. Looking specifically, because the hope would be not, again, we're not targeting in this case hot flashes or but we're targeting, can we actually help put myelin back on the nerves before they shrivel?
Yeah, and you'd probably want somebody on your team, right, who's like a breast cancer expert, and think about, you know, maybe ensuring that people are getting.
Yeah. They're the most
I would enroll in your study.
Yeah. Good.
Maybe I'm biased.
Come on back. No. Good, thank you.
You can fly me out there. It's like the NI.
Good.
All right, thank you very much. We'll move to the last talk for time. So if you could share your slides. So this is Dr. Anneke van der Walt from Monash University, who's co-leading this clinical trials group with me. And it is a study of crossover trial design, lessons learned from Botox-study for upper limb tremor in MS. Oh, I think you're muted. I'll unmute you.
Okay.
Okay, you're unmuted.
I can't see how I share my screen. Have I got, oh, there it is. Got it. Got it. Okay. So I'll just go back to presentation. Great. Can you see that?
Not quite yet.
Is it working?
No. Do you see the green box that says share? I don't think it's sharing yet.
Let's see. Let's go back to it. Sorry.
Okay.
Yeah.
Yes, perfect.
Right. Okay. So do you see the full screen, or maybe?
Yeah, you just have to put into slide presentation mode.
Okay.
Now we're seeing the presenter view actually. So
Sorry.
maybe do swap displays. I think swap displays should.
That's it.
Perfect.
Better? Great. Sorry. So I thought I would talk to you about experience that I had. This was actually a study that I did as sort of a side project during my PhD. But you know, it was a small crossover trial. And I think this is a trial design that we, you know, often see published and has been popular for a very, very long time. You know, it's often quite useful because of certain advantages. So we'll go through that. These are my disclosures. So a crossover trial design in its simplest form is really where you administer two treatments or a treatment and a placebo, and you give that consecutively to the same patient. There's usually a washout period between the treatments. And people are randomly assigned to either of the treatments when they enter the study. And then, so it's really sort of a repeated measures design where you have someone, you know, exposed, and you repeat the measures for each time period. So, so if you look at that graphically, if you have a person entering into a trial, you would randomize them to treatment A or treatment B in period 1. You'll measure those outcomes and then have a washout and have then another treatment period with the same measurements again. So statistically, if you think about it, you do have to consider this almost as a 4-period sort of design where you have, you know, time 1 and time 2 on this picture to the end of your first treatment, the washout period, and the start of your new baseline at time 3 and then time 4. So it's not just a simple pre and post comparison as such. And these statistical concepts are quite important to make sure that you are actually accurately interpreting your results. So the pros, of course, is that the patient can serve as their own control. So that limits you know, confounding variables like age and sex and social status and education and so forth. And because of that, you can often get away with a smaller sample size. So, and it works quite well in people with chronic conditions where you're not going to expect a sudden cure or a rapid change, of course. So it's often used in pain trials, neurological and behavioral interventions as well. The cons and the pitfalls is, of course, that you have to consider the order in which you're going to give the treatments very carefully, especially when you're using an active comparator. And the reason for that is that, you know, of course, if you give a treatment first with very dramatic side effects, even after the washout period, the person might be more sensitive to second time around or might be reluctant to continue and so forth. The washout period is also very important because you want to make sure that you can truly assess your second treatment period without any effects lingering from the first treatment period. And this really implies that you do have to know the dynamics of the treatment. And that can, you know, especially if you have a novel hypothesis, can be a bit tricky if you don't exactly understand how this could actually work, what's the duration of effects going to be, and so forth. So it requires some statistical expertise, as I said before. And the important thing is that we do have to try and separate the effect of the treatment from a possible period effect. So a period effect could be that, you know, if you do certain types of measurements, like say you're doing a cognitive test and you're repeating that quite often, you might be, you know, your patient might have learning effects, you know, regardless of which treatment they're getting. So they could look like they're getting better, you know, things like that you have to consider. So the period effect, outcome measures are very important. And then, you know, looking at, you do have to consider these as independent samples when you do statistical analysis, and looking at the change in an individual in the time periods and as your raw data, and then using those in your comparisons is probably, you know, a bit more of a valid concept. So with this study was basically started with observations that we had, where we worked with a very experienced movement disorder specialist who, Dr. Andrew Evans, and he, Botoxed a few MS tremor patients. And some of these people actually had dramatic improvements. So the thought was to see if we can, show this in a randomized control study, and this led to a crossover design. So basically, how the study was designed is that people were randomized to Botox injections or placebo at baseline. They were assessed 6 weeks after the injections, which we know we should see the peak effect. And then at the 12-week marks, they had another visit and they were crossed over here. So we know from generally that Botox is the biological effect should be gone by 12 weeks. Certainly from a pharmacodynamic response, that should also be the case. So the patients crossed over. And so overall, we had a 6-month trial without a washout period. So the clinical assessment, so this is basically what was interesting about this study was up until this point, studies of Botox in upper limb tremor, especially essential tremor, was really based on a very fixed injection strategy. So every person would receive the same injections, and that might be feasible in something like essential tremor, where the clinical picture is a little bit less heterogeneous than in MS. But as we all know, the tremor pattern in MS can be extremely varied. And so what we did is we basically identified the tremor pattern clinically and by EMG guidance, and then selected the best muscles for each patient with a top maximum dose of 100 international units per injections. And then to try and standardize our assessments, people had a standardized video assessment at each visit. And at the end of this trial, these visits, so each patient had 5 videos. They were all randomized and shuffled together, and they were rated by a completely independent movement disorder expert to get to the outcomes. So we had a look at our outcomes. So we mainly used a score called the Bain score, which is a score out of 10, which measures tremor severity. And we also had a few different things where we looked at different aspects of tremor: postural tremor, action tremor, pure intention tremor, rest tremor, and then also functional tasks. And we had to adapt scores from Parkinson's scores like the IPDRS, the CRST, and dysmetria scores as well, to try and come up with a battery of tests that would be appropriate. So we managed to recruit 25 patients with MS, of which 10 of them had bilateral tremor. And in those patients, we actually randomized both limbs separately. So we treated them as an independent arm. So people could receive, if they were bilateral, they could have received Botox in both arms at the start, or Botox and placebo, and vice versa, and so forth. So most of these patients had more advanced MS, so with an EDSS of about 5.5. And if you looked at the SARA score, which is score for the rating of ataxia and cerebellar dysfunction, they had more moderate to severe cerebellar dysfunction as well, which you would expect. But none of these patients had any upper limb weakness. So basically what we found is that after 6 weeks, and then also at 12 weeks, we could see an improvement in the overall tremor severity, and people also improved with regards to writing and drawing function. And then looking at the sub aspects of tremor. So if we looked at, postural tremor, both the severity and amplitude of that, action tremor, and so forth, we could consistently see an improvement over time that persisted up to the 12 weeks marks. And again, when we looked at functional tasks, we saw that, the ability to draw some straight lines improved. There was an improvement in pouring cups, and also in the 9-hole peg test to a degree. So So really the conclusion was, and if you look at this patient who has a right upper limb tremor, which is her dominant hand, she has sort of a fine postural and action tremor, which interferes quite markedly when she tries and writes. So the quality of the video is not that great, but hopefully you can appreciate how she's shaking over there. So we concluded that, you know, Botox does appear to help. So we can see 6 weeks after, she does actually has a complete ablation of that tremor without developing any observable weakness. And you can see this quite markedly when she's drawing and doing tasks as well. So this is actually, you know, now led to us doing a further study to explore this better. Because clearly we need to work out if what we observed, you know, can truly be, you know, can you really apply this to a whole group of MS patients, which is always difficult because of the variability that you see. So is this result really true? And I guess this is always where, you know, I think like Riley said before, you know, thinking about what reviewers would say and what did, you know, what we got told. So you do have to think about the washout period, and you know, we didn't do a washout period, and that I think you know, potentially could be a big problem. But because it turns out that in people with MS, with this type of tremor, in some people who do respond, we see a very prolonged effect. So some participants would have tremor ablation for 6 months, 9 months before it returned. So this is something to consider. And you also, you know, it's very clear that you do have to have an independent rater because of weakness, that if someone did get weakness, you know, if you were rating that yourself, it'd be very easy for you to pick up and know what treatment the patient had had, so you can unblind yourself quite easily. So we mediated a lot of the issues with the washout period, with this randomization of the videos and the independent rater. But certainly, you know, the lessons that I've learned from this has really informed our phase 3 study that we have almost completed, and maybe I can present that to you next year. But it has sort of, you know, emphasized a lot of different things to address in the study design, in the outcomes, and also what to expect. So I think that, you know, a crossover trial design has many advantages, and it can actually, you know, be a very good place to start. What we found with our trial is that it was hypothesis generating on multiple fronts. And using some of the strategies that Ellen described, we did sort of use standard of care MRIs to actually, you know, which we tried to schedule during this trial to get some structural information on the brain during this time, and that leveraged another piece of work, and again informed sub-projects and sub-studies that you can actually derive from a very small study, and that can then leverage a whole new program of work. So I think that, you know, starting off with a study like this could be very useful, and can be quite doable as well. But you do have to just be very careful designing it, and, you know, anticipating all the problems as far as you can. So I'll stop there. This is just the people who contributed to this particular study. And yes, happy to take any questions.
Thank you. I noticed that you had significant p-values, which with such a small sample size, which is, you know, impressive. And I wonder if maybe the crossover design helped with that when you have a small pilot study, if it is a short-lived effect of a medication or intervention that may, you know, be more powerful in a small sample size to detect effects. So I thought that was interesting.
Well, yeah, I guess that's the thing. But I suppose if you know, say you had Botox first and you had an improvement that persisted at your new baseline, and you then had placebo, which then may you know, you may actually, your tremor could get worse to the end of that second observation you know, period. So it's hard to, you know, so it could actually have strengthened some of the results or weakened the results as well. So I think it, you know, it can be quite hard to tease out, and it would vary quite a bit between, the responses vary quite a bit between patients. So, you know, and I think that just sort of emphasize that to truly provide first-class evidence, you know, you do have to go to a gold standard parallel design.
Cool, and we're working on with Bardia Nurbuchuk, who's at Hopkins now, a placebo or a crossover study of different fatigue treatments in MS. So that was another study where it may be applicable because the effects, you know, are pretty short-lived. So that's study is to be announced, but maybe he can present at this session at some point.
That would be great.
All right, thank you so much. Maybe if you stop sharing your slides, I'll just put our emails up again. Thank you everyone for joining. We're happy to have this first session for clinical trials, and we really, you know, are looking for speakers for future sessions. And so we'll send invites for that and invitations, but if anyone's interested, you can let us know whenever you have an interest for clinical trials, emailing me or Anneke with our emails up here, and they're on the IWiMS.world website as well. And then I know we're going to try to be off kilter by about a month from the epidemiology conferences, which is led by Dr. Mowry. So we're always happy to have people volunteer to give talks for these sessions.
As are we in Epi.
Yay. Thank you so much, Kristen.
Thank you.
Thank you for organizing.
Thank you.
Awesome
Thanks,
conference.
Kristen.
Good job.
Bye-bye.
Bye.
Great.